Welcome to G Fun Facts Online. Today we're tackling one of the most profound questions humanity has ever asked. Where did we come from? Not in a familial sense, but in a cosmic one. How did a sterile, rocky planet billions of years ago give rise to life? This question has shifted from the realm of philosophy into a hands-on, experimental science called prebiotic chemistry. Scientists are no longer just pondering the origin of life. They are trying to recreate it in the lab. We've made incredible progress, learning how the simple building blocks of life, like amino acids, could form from basic chemicals present on the early Earth. But the journey from a chemical soup to the first living cell is still filled with giant, unanswered questions. Today, we're diving into the 5 biggest hurdles that scientists face and the innovative new approaches they're using to overcome them. First, let's look at the challenges. The first major obstacle is what's known as the polymerization problem. It sounds complex, but the idea is simple. Life depends on long, chain-like molecules called polymers. Proteins are polymers of amino acids, and DNA and RNA are polymers of nucleotides. We have some good ideas about how the individual links in these chains, the amino acids and nucleotides, could have formed on the early Earth. That's a huge step, but getting those individual links to spontaneously connect into long, stable chains is another matter entirely. The problem is water. The chemical bonds that link these building blocks together are actually broken by water in a process called hydrolysis. This means that the very environment we think life emerged from, the primordial soup, was actively hostile to the formation of the very molecules life needs. It's like trying to light a fire in the middle of a rainstorm. So how did long, functional proteins and nucleic acids ever form and stay stable? This is a critical puzzle that researchers are working on, exploring how minerals or cycles of evaporation might have provided a safe haven for these chains to grow. The second massive challenge is the origin of homochirality. This is one of the most fascinating and mind-bending problems in all of science. Many of the essential molecules of life are chiral, which means they exist in 2 forms that are mirror images of each other, like your left and right hands. They are made of the same atoms, but they are arranged differently in space. In the chemistry of life, this distinction is everything. All the amino acids used in proteins on Earth are left-handed, while the sugars in DNA and RNA are right-handed. There's no known functional reason why it had to be this way, but life is incredibly consistent about it. The problem is that whenever scientists create these molecules in the lab using prebiotic methods, they always get a perfect 50-50 mix of both left-handed and right-handed versions. This is called a racemic mixture. So how did early life select only one-handedness? What mechanism filtered out all the right-handed amino acids and left-handed sugars? Was it a chance event that got locked in? Or was there some fundamental force or environmental condition, perhaps involving polarized light or specific mineral surfaces, that favored one hand over the other? Solving this would be a monumental breakthrough. Third, we have the information enigma. This is less about chemistry and more about the birth of meaning. A polymer like RNA is just a string of chemical units. How did such a string first come to encode meaningful, functional information? Think of it like this. If you spill a bag of alphabet soup on the floor, you get a random sequence of letters. You don't get a Shakespearean sonnet. How did a random sequence of nucleotides on the early Earth transition into a functional molecule, like a ribozyme that could speed up a chemical reaction, or even a self-replicating molecule that could pass on its own sequence? This is the software problem of the origin of life. It's the leap from random chemistry to a system that carries and processes information, a foundational requirement for evolution. Bridging this conceptual gap is one of the most difficult challenges for researchers. Fourth on our list is the rise of the protocell. Life needs to be contained. The complex chemistry of life has to happen in a controlled environment, separate from the chaos of the outside world. This is the job of the cell membrane. It's relatively easy to show how lipids, which are fatty molecules, can spontaneously self-assemble into hollow spheres called vesicles in water. These are our protocells, the earliest ancestors of modern cells. But a protocell has to be more than just a passive bubble. It needs to be able to capture building blocks and energy from its environment, use them to grow, and then, crucially, divide to create daughter cells. How did these simple lipid bags develop the machinery to manage an internal metabolism, to grow in a controlled way, and to replicate? Recent experiments have shown that protocells in the lab can be made to grow and divide, which is a huge step forward. But linking this to a stable, information-carrying molecule inside is the next frontier. Finally, the fifth major challenge is reconciling competing theories. For decades, the field has been dominated by two major camps. On one side, you have the RNA world hypothesis. This theory suggests that life began with RNA, a versatile molecule that can both store genetic information like DNA and catalyze chemical reactions like proteins. It's a compelling idea of an all-in-one molecule to kickstart life. On the other side is the metabolism-first hypothesis. This camp argues that self-sustaining cycles of chemical reactions were the first form of life, and that informational molecules like RNA came along later. Think of it as the kitchen coming before the recipe book. Both theories have strong points, but both are incomplete. The major challenge for the future is to find a way to integrate them. Perhaps the answer isn't what came first, but rather how did metabolic cycles and informational polymers arise and evolve together, codependently from the very beginning? So, with these 5 giant hurdles, you might think the problem is unsolvable. But science thrives on challenges, and the path forward is illuminated by several powerful interdisciplinary approaches. The first, and perhaps most important, is the rise of systems chemistry. Instead of looking at one reaction at a time, this approach tries to understand how entire networks of reactions can work together to produce complex, lifelike behavior. A brilliant example is the cyanosulfidic chemistry pioneered by John Sutherland's group. They showed how a single set of starting materials, based on hydrogen cyanide and hydrogen sulfide, when exposed to ultraviolet light, could produce the building blocks for not just one type of molecule, but for nucleotides, amino acids, and lipids simultaneously. This suggests that the ingredients for all the major components of a cell could have been created in the same place at the same time from a common chemical origin. It's a radical shift from the idea of separate, isolated pawns for each type of molecule. Another key trend is exploring messy chemistry. Early experiments famously used pure, refined chemicals in highly controlled lab settings. But the early Earth wasn't a sterile laboratory. It was a complex, messy, and chaotic place. There was a jumble of different minerals, chemicals, and conditions. Modern researchers are now embracing this complexity, running experiments in heterogeneous mixtures that more closely mimic a primordial puddle or a volcanic vent. They're finding that this messiness isn't always a problem. Sometimes, other minerals and chemicals can actually help catalyze important reactions. We're also realizing the critical role of environmental dynamics. The early Earth wasn't a static environment. It had day-night cycles, fluctuating temperatures, and, most importantly, wet-dry cycles. Imagine a shallow pool of water on a volcanic shoreline. It's full of simple chemicals. The sun comes out, the water evaporates, and those chemicals become highly concentrated. This concentration could be the key to solving the polymerization problem, forcing molecules together to form chains. Then, rain comes, the pool fills up, and the new polymers are dispersed. This cycle, repeated millions of times, could have been a powerful engine for generating chemical complexity. Simulating these dynamic conditions in the lab is now a major focus of research. Of course, we aren't just limited to studying the Earth. We have clues from outer space. The study of meteorites provides a direct window into the chemistry of the early solar system. Scientists have found amino acids, nucleobases, and other complex organic molecules inside meteorites, like the Murchison meteorite. This provides powerful evidence that the basic building blocks of life might not have needed to form on Earth at all. They could have been synthesized in space and delivered to our planet during the early heavy bombardment period. This is called panspermia, and future missions to collect samples from asteroids and comets will give us an even clearer picture of this extraterrestrial chemical factory. Finally, we have the immense power of computational chemistry. With modern supercomputers, scientists can simulate prebiotic reactions with incredible accuracy. They can explore thousands of potential reaction pathways to see which ones are most plausible under early Earth conditions. This saves countless hours of lab work by helping to guide experiments. It allows us to explore the vast, uncharted territory of chemical possibilities and identify the most promising avenues for creating life's precursors in the lab. The quest to understand the origin of life is far more than an academic exercise. It is a fundamental part of the human journey to understand our own existence and our place in the cosmos. Every small breakthrough, every new reaction pathway discovered, brings us closer to answering that question. This knowledge doesn't just look to the past, it guides our future. By understanding these specific conditions that allowed life to emerge here, we get a much better idea of where to look for life elsewhere in the universe. We can identify potentially habitable exoplanets and design missions with instruments tailored to look for the specific biosignatures we predict. The road ahead is undoubtedly long. Many of our current theories will be revised, and some may be discarded entirely as new evidence comes to light. But that is the nature of science. The relentless spirit of inquiry that drives this field ensures that we will keep pushing the boundaries, venturing deeper and deeper into the mystery of creation. And when the answers finally come, they will forever reshape our understanding of chemistry, biology, and what it truly means to be alive.